and welcome to today's webinar. My name is Melissa and I'm a Scientific Training Officer in the training team at EMBL-EBI. I will also be your host for today's webinar. In today's webinar, we're joined by Emily Vanolo and Enzo Medico, and they're here to talk to us about EurOPDX, the Data Portal, a repository of patient-derived xenograft models. So with that, I'm going to hand over to Emily to kickstart the webinar.
Thank you, Melissa, and thank you all for attending our webinar. We're happy to present the new EurOPDX Research Infrastructure, and in particular our EurOPDX Data Portal, the new repository for patient-derived xenograft models. So I will start with this introduction, and Enzo, the coordinator of the project, and the research infrastructure will continue with the demo of the functionalities of the data portal. So first of all, you might not be all fully familiar with patient-derived xenograft models, which we will refer to by PDX models. They are obtained by implanting tumor fragments of patients into immunodeficient mice, letting the tumor grow into that mouse, and upon reaching humane endpoints, sacrificing the mouse and reimplanting the tumor pieces into other mice to obtain a second generation or passage, etc., etc. So at each different passages, the PDX tissue is biobanked as FFPE, fresh frozen and cryopreserved tissue, and a series of and a series of validation and characterization of the model is performed by histology, by fingerprinting to verify the genealogy of the samples, by sequencing, and also drug dosing studies. Among characterization that can be done. So the PDX models keep the features of the original patient tumor and are now widely recognized as clinically relevant preclinical models. There have been established, a lot of models have been established in Europe and worldwide for a lot of cancer types. But they are time-consuming and costly to establish and mostly to characterize. So back in 2013, it became important for several research teams in Europe to gather in a network and integrate the collection of established PDX models, first of all to perform collaborative projects, but mainly to be able to perform studies for genotype response correlation on a population scale rather than on a couple of models, and this way to increase the predictability of preclinical data for success in the clinic and improve oncology development success rates. So as I said, in 2013, we established the EurOPDX Consortium as an initiative to promote scientific collaboration in PDX research in Europe, and beyond, because we have a member in the US, integrate available PDX collections, and also raise standards in the field. And a couple of years ago, the consortium partnered with additional institutions and academic institutions and SMEs in Europe to on an EU grant, a 5 million EU grant, which began in February 2018, and under which we are building the new EurOPDX Research Infrastructure to allow PDX research and opening access to PDX models, expertise and resources of the consortium for academic researchers and industry. I will go into the details of how of the services the research infrastructure offers to the researchers under the project. And so the main objectives of the project are focused at providing those services to the scientific community. And first of all, that is the main topic of our webinar today. We aim to develop and employ efficient and user-friendly IT tools to capture information from the established PDX models from the EurOPDX members, so that metadata, but also clinical, molecular, and pharmacological annotations, integrate those PDX data into a virtual repository, that is our EurOPDX Data Portal, and enable efficient search, analysis, and visualization of the PDX data for users of the EurOPDX Research Infrastructure to select their model of interest and generate hypotheses on the data. The building of such a research infrastructure, so this is a distributed infrastructure with 6 nodes that perform the experiments. This requires harmonization and standardization of procedures. Actually, this was recognized as an important topic in the field already years ago when seeing the lack of reproducibility of data, of preclinical data. So we've been working in the first 18 months actively at standardizing procedures across, in particular, across the nodes of the research infrastructure for the biobanking of models, their propagation, and to allow their efficient exchange and transfer across institutions, and for the performance of drug efficacy studies. And so finally, as I was saying, all of this work is important in the field, but even more for the provision of free access by the research infrastructure. And I will go into the details of the 3 types of access possible, which are sample, deposit, and test. In terms of standards, it all started several years ago with the definition of the minimal information that need to be captured for PDX models. We had started at draft standards within the EurOPDX consortium, and this was consolidated, led by the EMBL-EBI in Cambridge, with a global participation from the PDX field in the US in particular and worldwide. And this allowed to agree on all the clinical PDX model and validation data that needs to be captured for a PDX, for an established PDX models. So this was published in Cancer Research in 2017, and this was the basis for building the repository, the EurOPDX Data Portal. And this is also a basis for our standards in general, because so among the standards we are working on, sorry, I'm looking for the mouse, but it's not working. So the standards we are working on relate to the validation and the quality control of PDXs, and in particular the health monitoring of models. So there will be a systematic health screening of models to be able to provide this information to the users receiving the models, or for whom we are using the models, because there might be some pathogens that are there that can be recognized as not having a big impact, but still they can be, like C. bovis or the LDEV virus. And together with the minimal information that I mentioned, we will include all this information in a PDX passport. So each PDX biobanked under the EurOPDX Research Infrastructure will be accompanied by this PDX passport with full information about the clinical data, the validation of the PDX, including health monitoring. We've been also working on SOPs for biobanking and in vivo drug efficacy studies, as I mentioned. And also, as an important part of harmonization of procedures, the same LIMS system, which is called LAS for Laboratory Assistance Suite, that was developed by the University of Torino, has been installed in all 6 nodes. So this allowed to further increase interoperability between the nodes of the infrastructure. So all the standards, so there are a few of those, this information that is already included in the dedicated web page of our website. And all the different SOPs that we have just finalized will be included in the next few months. So An important aspect of our activity is to promote the access of these PDX models to the scientific community. So we call it transnational access. This is a European Commission word, but basically it means that we are organizing the free of charge access to the PDX resource and the expertise of the consortium for academic mostly, but also for industry researchers through one of the 6 nodes. So the 6 nodes are showed in the map on the right. And actually, so we are organizing this webinar in the frame of the second call for access to the models that was launched at the end of September and is open until the 22nd of November. So you can access models from the infrastructure through 3 different access types. The first one, we call it sample, and that it allows the shipment of cryopreserved PDX tumor samples to selected academic laboratories. So shipment of models is only available for academic labs. So all of this access is organized as call for proposals, as I mentioned, and selected by an independent committee. So when you will have chosen one or several PDX models in our data portal, like we will show you in a few minutes, and upon selection by the committee, we will be engrafting and growing this model in NSG mice according to the agreed SOPs, generating aliquots, doing all the necessary quality control as also described in our SOP and agreed on our SOP, and shipping the models to you for your experiments together with the PDX passport corresponding to these models. Another way of accessing models is through depositing them to the research infrastructure. So say you have very interesting models, very rare models that you would like to be more visible to the scientific community. You could apply to have them deposited by first having them biobanked the same way, so health checked and then engrafted and expanded through the same SOPs in NSG mice and stored in one of the 6 nodes, and also visible in the data portal. So we would work with you at getting all the associated metadata and molecular data if you have characterized these models. Finally, a third way of accessing the models is what we call tests. It's the performance of in vivo drug efficacy studies on selected models. So this test access is also available to biotechs and of course to academic labs. So you would similarly to sample be able to select the models you want to work with in the EurOPDX Data Portal and provide the drug to one of the 6 nodes. And the drug efficacy study would be performed in one of the 6 nodes. So you receive at the end all the final report with the experimental results and the quality controls performed. So again, our second call is ongoing. There will be other calls in 2020, but you may want to apply right now. And by the 22nd of November, all information and document is available on the dedicated webpage of our website as well. And I will provide the email contact email for any question later. The only document that is missing is for the test access. It will be available within a few days. And if you send us an email, we can send it to you directly upon when it's available. So before we go to the demo, I just want to add a few words about the EurOPDX Data Portal. So you have the address here. It's been developed by project partners, and in particular the EMBL-EBI in the UK and Masaryk University in Brno in Czech Republic, as well as the University of Torino. And the first version was released at the end of July 2019, so a few weeks ago. It's powered by the PDX Finder, which was developed by the EMBL-EBI in collaboration with the Jackson Laboratory on another grant, on an SCA grant. So we are reusing the functionalities of the PDX Finder and using their templates to collect the data from the EurOPDX partners, as well as all the filtering options we will show you in a few minutes. The additional functionalities included so far are the fact that we included a user authentication service to provide users with the dashboards, but also for some tracking purposes due to the EU grant. What we have been working on as well is linking the data in this repository to CBio portal whenever molecular data are available. So those data are available for colorectal cancer at the moment, and we are working at adding more. So there should be some breast cancer data available in a few days. And later in the project, we will also link to a 3D visualization software that has been developed by a small SME in Italy called Genome Cruiser, which allows to visualize molecular data in 3D. What we also added in the PDX Finder is the way to filter the models for access. Indeed, so in our EU grant, we are mainly focusing at providing transnational access under the grant to the PDX models, but all this activity is also in general to promote collaboration within labs, avoid duplication of efforts, and increase the visibility of the established PDX models. So some of the models, this is not the majority now, but some of the models might only be available on a collaborative basis. That means that you would need to contact the owners of the PDX to try to establish a collaboration. So we'll present in detail how you can browse the data in the EurOPDX CBio portal, and this is just an illustration of the Genome Cruiser, which will be available later in the project. So for visualization, 3D visualization of the molecular data. And so finally, where are we in terms of including PDX data into the EurOPDX Data Portal? So as of today, for this second call, we have almost 900 PDX models. You have here the distribution of those models. So most of them are colorectal cancer models, but you have also a nice collection of gastric cancer and breast cancer models, as well as 10 pancreatic cancer and 4 non-small cell lung cancer models. Those models are associated, in addition to metadata, with a certain number of additional characterization data. Cytogenetics are mostly immunohistochemistry, like for breast, in the first line for breast cancer models at the Institut Curie in Paris. 3 markers include ER, PR, and HER2. There are also mutation data and copy number alteration data. We also included already patient treatment data for some of the models and drug dosing data in PDXs, in particular cetuximab information, cetuximab treatment in the colorectal cancer models. So we are, of course, actively working at including more PDXs and more data, even for the PDXs that are already there. We are always available to ask. I mean, if you have any question of those models, please feel free to contact us through the email I will provide later. And in a few weeks, we will also have, first of all, some ovarian cancer and endometrial cancer models. Yeah, so this is the email, ta@europdx.eu, but I can switch to Enzo now. Enzo, if you are ready.
Yes, thank you. Hello, everybody.
And
this is Enzo. Now we are going to demonstrate the functionality of the data portal and of the associated CBio portal instance. Emily is going to show videos just to avoid problems. I have a poor connection from where I'm talking, so as you can see here, we are in the data portal website. You can see the address, the URL, and there is one tab that you can click, which is the Explore the EurOPDX Data. This will bring you to the data portal. You have to register and log in. Of course, I had already registered. When you log in, typically the typical choice would be, for example, to use Google, to sign in with Google, but there are many options. So once you choose your option, you finally log in and enter the data portal. Now we are switching to the next video. Here we are in the data portal. There is a search field, but the most interesting part is the filter tabs that you can use. So for every type, you can see you can select data that have certain types of information you are interested in, or the source, so the single data set provided by the various centers. The access modalities, so samples that are available for transnational access or for collaboration. You can search for specific gene mutations if it's included and the type of variants you're interested in, or again, a gene that has copy number alterations. And there is a dedicated panel for breast cancer biomarkers. You can understand if the patients have been treated or not, if they are naive or not naive. The type of treatment of the patient, and when available, drug response of the PDX model. And then here is the type of cancer. Of course, here is the larger collection. As Emily said, now we are concentrated on a more limited number of tumor types. Again, basic things like gender and age bins, where you can select tumors in younger or older patients. So in principle, you have a wide spectrum of possible filters to select the model of your interest. Now I'm going to present one possible way of selecting model through the data portal, in particular focusing on breast cancer. So we can start the next video. So now we try to concentrate, for example, on searching models for which we want to explore HER2 amplification and gene mutations available. So we can try to select from, for example, from the data set, say, I want samples for which there are gene mutation data. And we see we have more than 600 cases with gene mutation information. And then we select, like, for example, HER2, ERB2 positive, okay? And when we come to this very selected system, we have 3 cases which have HER2 amplification and mutation information. Now let's say, well, I want to know in these genes which have P53 mutated. So we type, you see there is a system for helping you select the gene of interest. And there are a number of variants available, models with many different variants. And we just select all because we don't know what type of variant is included in the 3 models of interest, and you pick search. And out of this search, you can find one model that has P53 mutation and HER2 amplification. You click on the model ID, and in this way, you can see all the information available for that particular model. So the patient of origin, the age, the histology, the tissue, the grade, the stage, and the type of mouse in which it has been implanted and propagated. This is the basic information from the model, information about the patient of origin, and information about molecular data. So this is a link to directly see when are available mutation data from this model. So in this case, when you click here, you see all the mutations that have been detected in this model. So in addition to the one of interest, other things could actually be additional mutation that could be of interest. So in this way, through a number of criteria and annotation of the models, you can find one or more PDX models of interest for a specific project. I think we are close to the end exactly of this video. Okay, so this is one possible approach. The second is you can open but not launch the video. The second option is to use the data portal to select a subset of samples and build a CBioportal view of these samples, so including molecular data that are available. I'm going to show this for colorectal cancer in this case. So we can start the video. So now in this case, for example, we want to take one entire study like data source. We can take the Candiolo Cancer Institute colorectal, click here, and it comes out that we have 600 models from this study. So it's a big study of colorectal cancer, a big set of models. And if you scroll down, you see on the bottom, export to CBioportal. You click this button, and this brings you to another page, which is going to, where you can track you click export data. So this is the description of the data set. And now you see a bar that is building. It takes some minutes because the system is building a CBioportal instance, so it's creating the instance. The video, I think, stops here and then continues once you finish the process and all the procedure has been completed by the system. The page changes. Now you have a green button with CBioportal, and you can click it. And now when you click it, you open the CBioportal for the 639 samples of interest. There is some disclaimer. You just go through the disclaimers. And here we are in the EurOPDX CBioportal. Now we built this study, IRCC, and you can select information of interest. I think the video is almost done here because there is then one video dedicated to the use of the CBioportal to identify and select samples. And this is the last video. So now I have already written down genes of interest. This is particularly useful if you want to select genes of interest which are non-obvious. In this case, for example, I wrote IGF2, RB2, and now you can see you just type the gene and there is a system that runs a check whether the gene symbol is okay. All gene symbols are valid, so you submit the query. And now we have the oncoprint, so the aggregate view of all the models. You can see how many samples you have per patient. In some cases, there are more PDX that have been profiled, even up to 3. And there is a color code for every sample. Every sample is a column, like IGF2 mutation. The red means amplification, or missense mutations are in green. Here you can see when you mouse over one of these features, it displays the information that is available. Okay, like in this case, there is MYC amplification, and so on. Like there is one sample here where you have RB2, which is mutated, copy number amplified, and mRNA upregulated. So this is a sample, for example, where RB2 is really high, really strong, mutated and amplified and overexpressed. You can, in this case, just by the oncoprint, go to identify the sample. This is the sample ID. You select, click on the sample ID, and in this way you can open the page from CBioportal with information about the sample. So here you can see this sample has a total of 67 mutations. So of course, in addition to the ERB2 mutation, there are other mutations, and they are all reported here. This sample has only one copy number alteration described, and then you can click on clinical data and see the attributes. Also from here, you can see information available from the samples, the age, and other information from the sample of origin and from the patient. So this is one way, directly from the OncoPrint. Another way is to use the plot function of CBioPortal. Here in this plot, can you stop it a little bit? Can you stop the video? It's going a little bit quick, and I need a moment to explain things here. So here on the x-axis, you don't see, but you see three categories: gene loss on the left, gene intermediate, normal copy number, and amplification on the right. And on the y-axis, we have mRNA expression of the gene of interest. You can restart the video, thanks. And here you can see that when you mouse over each individual gene, you can see details. You can change the gene of interest, like ERB2. When you click on ERB2, you can see there are some samples which have ERB2 amplification and overexpression. The green one also have a mutation. So you can directly select samples from the plot and do the same thing. When you mouse over, you will see information for each. This is MIC, and you can select one model of interest just by mousing over and clicking on the sample ID. And again, you open the page with all the information related to that sample. So in this case, again, this sample has a total of 92 mutations and copy number alterations, not only one, there is a second copy number alteration. Interesting is MAD4 deletion. So this model also has the TGF-beta pathway genetically inactivated, which could be maybe of interest, depending. So this is another way of selecting models rather than based on their annotation, which is typically done on the data portal based on molecular profiles and molecular features, gene expression, copy number, and mutation. This is a typical approach of CBioPortal. So if you are familiar with the CBioPortal of the Memorial with the main website, we are not doing anything different at this stage. There are profiles for mutations. For example, if you select ERB2, this is the distribution of mutations in the models of the entire colorectal cancer dataset. So you can browse here and select the mutations of interest, for example. This is another way of finding information, aggregate information on a data set and identify interesting model. Now, this of course has been done for the usual suspects, so I put ERB2, but if any investigator has a particular gene of interest, that gene of interest can be explored in the collection to find models that have alterations or deletion or amplifications or overexpressions. There is a lot of information that simply pops out from the mutation in terms of the functional consequences. And I think we are finished. You can run a mutual exclusivity analysis, for example, just to see whether these alterations tend to co-occur. Now, in this case, these three alterations do not have a significance co-occurrence. So there is a tendency to co-occurrence or to mutual exclusivity, but if you just select significant only, you will see that nothing comes out. So there is no significant either mutual exclusivity or co-occurrence of these alterations. And this is one classical way of exploiting the CBioPortal for complex queries on molecular profiles. So the video is finished, and so is this demo. So clearly, if any of you wants to have more information, there are how-tos on the website. So there are guidelines published, and you can always ask us. Okay, you can also always ask us if there is any need to further help with finding the models of interest. And with this, I am finished.
Yeah, thanks. And so I was just showing the tabs in the data portal where you can find information on the tools and services, on the CBio portal, on the registration if you're having any issue. And you should not hesitate to contact us through the IT email, or mostly if you're planning to apply through the transnational access call. Please use the TA@EurOPDX.EU email, and we will help you in the application process or selection of samples. We are also, in any case, looking at the selection with the selected users upon selection, by the external committee. So we would verify in the collection that the models you picked are the best suited for your project. So the deadline of the current call is 22nd of November, and we will add continuously more models and data as we said, and there will be other opportunities to apply in 2020. With this, and you also have the address of the website and the Twitter account if you want to follow us on Twitter. With this, I guess we can take questions through the chat, if there are any.
So if you do have a question, please do write it in the chat box. While you're doing that, so just a quick reminder that this webinar is part of our regular Wednesday webinar series. The next webinar in the series is on the 13th of November, and it will be focusing on the single cell atlas, which is being developed here at EMBL-EBI. You can find lots of information about our upcoming webinars and other training that we offer on our website, which you can see on the screen there now. Okay, so there don't seem to be any questions this afternoon. However, if you do think of a question, please do feel free to get in touch with the EurOPDX team through the links that they've just shown you. So
yeah, I see some familiar names in the attendees, so they already knew how to contact us, so please don't hesitate.
Okay, so that just leaves me to thank Emily and Enzo once again for a very interesting webinar, and to thank you all for joining us this afternoon. We hope to see you again in another webinar, and enjoy the rest of your day.
Thank you all.
Thank you
Melissa.
very much.